So, is there any student from my class that's here? No one came. Oh, I'm gonna have to talk to them. So I told them, you guys should come. It's a nice marriage of sex hormones and sex differences and how that interacts on the appetite and feeding systems. And he's also embraced the parts of how endocrine disruption affects overall signaling in this process, looking at it in development, in adults. Really interesting work. So I've known Troy over the years, gotten to know him through the Society of Behavioral Endocrinology first, and then he, like I did as well, transitioned into the more endocrine disruption part. So we've kind of been in parallel paths here. So, but his work is really fantastic, and it's up, which is good. The title of his talk today is Interactions of Organophosphate Flame Retardant Exposure and Diet on Energy Balance in Mice. And I will turn it over to Troy after I turn on this light.
All right, thank you, Chris, for that introduction. Welcome, everybody. Thank you for coming. As he said, this is one of the areas that I focus on in my lab, and it's really at the intersection of metabolism, neuroscience, neurophysiology, and endocrine disruption. So, just for background, if you don't know what an endocrine disrupting compound is, it's basically a chemical that can act like a hormone or can mimic hormone actions, inhibit hormone actions, interactions with the receptors, interfere with their production, etc. So the type of endocrine disruption I focus on is where the EDC is interacting with the receptor, either interfering with the receptor signaling or initiating receptor signaling in a disruptive manner. Some common EDCs that a lot of people probably know about are like bisphenol A, BPA, which is in plastics, genistein, which is a phytoestrogen from soy, 17-alpha ethinyl estradiol is part of the pill, the contraception, right? So to a person taking EE2, it's not an EDC, but if you urinate it out into the wild, and then it becomes an EDC to fish and everything else in the wild. So what I'm focusing on is our flame retardants. So flame retardants, we're basically setting in a soup of flame retardants as I speak right here, because regulations have it that all the carpeting and the fabric on your seats and the walls and stuff all have to have some sort of anti-combustive material added to it to reduce flame, right? Flame retardants. And so previously, these flame retardants were called polybrominated diphenyl ethers, or Pbdes, but they were found to have neurological effects, endocrine effects, reproductive effects, a whole range of effects, especially during sensitive developmental time periods. So they were largely phased out in the mid-2000s, about 10, 15 years ago, and replaced with organophosphate flame retardants. So there's a common mixture called FireMaster 550, and it's a mixture of multiple different types of these OPFRs that we are now being exposed to every day. So the common exposure, of course, in your home is like you have it in your furniture, electronics, carpeting, drapes, baby toys, all over the place. So it's usually ingestion, inhalation through house dust, ingestion, especially for infants or neonates. They like to put everything in their mouth, and so they're suckling on it, so they're constantly being exposed. Again, it's pretty much found everywhere. You know, I flew here, as you would expect, airplanes are chock full of flame retardants because you don't want to blow up midair, right? So some of these flame retardants, the ones I'm going to be working with, I'm going to say TDCPP or TPP or TCP, are pretty common, and they actually have been shown to interact at some concentrations with steroid receptors, androgen receptors, glucocorticoid receptors, and my special receptor, estrogen receptors. So that's why I chose these and why I'm targeting their actions. So most studies are, when they do these EDC studies, it's a developmental exposure. They give it to the dam, and then they look at the fetus and the offspring, or they expose the neonate, and then they look at what happens. Well, I'm interested in what happens throughout the lifespan because we are constantly exposed to them. So I have some projects looking at maternal and neonatal exposures as well as adults. So the stuff I'm going to talk about today is all my adult work. Because while toddlers and babies are exposed, but we're also exposed to house dust, drinking water, offices, and aircrafts, right? So here's the structure of the Pbdes. These are the ones that are largely phased out. Of course, there are many different variations depending on how many bromides you have to are attached. Here's just for comparison, estradiol structure, and here's the compounds that I've chosen. Now, some of these compounds you can see, oh, well, you can see how this looks like an estrogen because it has these benzene rings. But one of the things that people investigate is our body metabolizes these compounds into more bioactive compounds. So instead of detoxifying and making them safer, it actually makes them more of an endocrine disruptor. So if you knock off one of these rings, you're going to have DPP, and that's going to make it even more accessible to the estrogen receptors and interact more with the receptor. So while we're administering these compounds, the effects may actually be coming from their metabolites. So just some data showing that overall flame retardants have been shown to interact with classical estrogen receptors. There's a whole range of literature reviews out there about it all. In fact, metabolites, as I said, of Pbdes, which is one of the most common ones, is quite efficient at targeting estrogen responsive genes in mammals, fish, and in chicks. Methylated Pbd47 also can be anti-androgenic, so it's not always acting through estrogen receptors. In terms of OPFRs, not as much is known. And until just recently, there's only been a few studies in fish and a few studies in chicks showing that it can interact with ER alpha and beta. It can interact with other nuclear receptors as an ER antagonist. They can also interact with PPAR gamma, peroxisome proliferator activated receptors. I don't get that right. Right. So, just the comparison with PBDEs. So these are largely phased out, but see some of these effects that includes thyroxine, thyroid hormone, affinity for multiple receptors. It can cross the blood-brain barrier in the fetus, disrupt thyroid hormone signaling. They've been shown associated with decreases in learning and memory and motor function. OPFRs, there's less known, but in some animals, fish and chicks, we've shown to alter hypothalamic gene expression. Again, some potential affinity for nuclear receptors, thyroid disruption in fish, and there's been a few studies showing that like TCBP can cause seizures in adult female rats, but that's at really high doses, like doses that fire people are exposed to, not necessarily what you're exposed to studying here. So what I was interested in is, what if we just expose some mice? At the time, there were no mouse studies when I started this project, and just adult mice, and look at gene expression in the arcuate, which was my area of interest. So just sort of give you an idea of why I'm interested in the arcuate nucleus. It's this nucleus that's right above the stalk that leads to the anterior and posterior pituitary. So at this area, it's at a leaky section of the blood-brain barrier, which is good because that's how signals from the periphery act on the neurons in the arcuate nucleus. But it's also bad because if chemicals get in your blood, they're going to be interacting with these neurons in the arcuate nucleus. And again, since it responds to circulating signals such as insulin, which controls sugar, leptin, which comes from the fat, ghrelin, which is the hunger hormone. So we're interested in how it may interact with these receptors or these hormones, including ER alpha and PPAR gamma. The reason why, and this elegant circuit that Chris was talking about, is why we're looking at these arcuate neurons is because we have neuropeptide or AGRP, or GUDI-related peptide, neuropeptide Y, AGRP neurons, and that they interact with POMC neurons, or pro-opiomelanocortin neurons. And pro-opiomelanocortin neurons, or POMC neurons, suppress food intake, increase energy expenditure. MPY neurons do the opposite. They increase food intake and suppress energy expenditure, right? So it's not this nice little circuit. They're kind of considered sort of like at the headwaters of a lot of the metabolic processes controlled by the brain, because they receive all these signals, including ghrelin from the stomach, PPY, leptin from fat tissue, insulin from the pancreas, etc., and a whole range of other signals, including steroids. All right, and they all signal to other areas of the brain, specifically in the hypothalamus, to turn on other neurons that are then going to control feeding behaviors, control energy output, brown adipose tissue activity, etc. So a whole range of actions. So my basic hypothesis is that OPFRs are going to interact with ER alpha and PPAR gamma, which are expressed in these neurons, and alter the physiology, the gene expression, the neurophysiology of these neurons, which then, or their responsiveness to these hormones, and then will alter downstream activity in the periventricular nucleus is one area that they innervate, and that's going to turn on or turn off energy intake or energy expenditure, and you can have disruption of energy balance. Now, no one has ever shown this sort of stuff with endocrine disruptor compounds. And it's one of the reasons why I went from studying sea urchins to studying guinea pigs and mice, is because I had an interest in EDCs. I'd shown effective EDCs in sea urchin development and potentially this non-genomic estrogen signaling. So I went to one of the leading experts in the world on estrogen signaling, rapid estrogen signaling in the brain, and ended up doing a postdoc with him so I could learn electrophysiology and all these techniques that I wanted to then apply with endocrine disruption, because no one was really looking at the neurophysiological effects of endocrine disruption. So to do that, we just did a straight up exposure study and then looked at gene expression in the arcuate. And this was the master's thesis for Elizabeth Crum, where we took adult male and female mice and exposed them for 28 days to a mixture of the OPFRs, either at 1 or 10 milligrams per kilogram per day each. So each one of these was at 1 or 10 milligrams. A positive control of ethinyl estradiol, and then an oil vehicle control. The males were intact, and because I didn't want to have interference with estrogen signaling on these estrogen responsive genes, I overectomized these females before we dosed them. So here's POMC, MPY, AGRP. CART is co-expressed in POMC neurons, so cocaine and amphetamine regulated transcript. It's also involved with energy balance. It's also regulated by cocaine and amphetamine, but that's not what I'm interested in. So what we, to my shock and surprise, we saw that POMC expression was increased in males with exposure to the OPFR mixture and slightly suppressed in females. CART was suppressed in males and females. MPY was suppressed in males, and AGRP was increased in females. We went over this data like 5 or 6 times because I didn't believe it, because I didn't expect anything to happen, because everybody's always like, oh, it's always a developmental effect. There are no effects of EDCs in an adult exposure. What are you doing this? And so we were like, well, something's going on here. This is exciting. It ended up getting my first NIH grant, so it was very useful. I'm very indebted to Liz for her work. So this profile, though, is very interesting, though, in terms of energy balance, because POMC and CART suppress feeding, MPY and AGRP increase feeding, but they're differentially regulated in males by these EDCs. So if you have an increase in POMC and a suppression of MPY, you would think, oh, well, that's going to decrease feeding, actually, which is opposite of what you think of a lot with EDCs, is they can be obesogenic. But then you also have an effect on AGRP, which AGRP is an endogenous antagonist to the receptor for one of the protein products of POMC, alpha MSH. So it interferes with the receptor, blocks the receptor, and blocks alpha MSH signaling. So this is sort of like a mixed neuropeptide profile. So what's going to happen in the males? We don't know yet. So we repeated this experiment again, which I'll talk about in a second. So we also looked for leptin receptor, insulin receptor, ghrelin receptor, ER alpha, and PPAR gamma. And we saw a lot of sex-specific effects. We saw big increases in the males and very little in the females. There were some effects with insulin receptor. PPAR gamma was increased in both males and females, but ER alpha was suppressed in the males with no effect in the females. Which is interesting because in females, if you give estradiol, it's going to suppress ER alpha expression. So my idea is like this wasn't necessarily directly through ER alpha signaling because there was no effect in females on ER alpha. It could be through PPAR gamma, which is what we're seeing. I'm interested in potassium channels. I specifically I've never thought I'd be considered the world expert on the M current or the KCNQ channels in the hypothalamus, but I am, because I have like now 8 publications on it. It wasn't a goal as a little kid that I was like, I'm going to be, but there you go, right? So this is the channel I usually study, the M current, and we saw that OPFR exposure altered the expression of KCNQ subunits, specifically in males. So again, a lot of sex-specific effects. So the more M current you have, the less excitable your neuron is going to be, right? So the M current it's an inhibitory. So if you have more of it, your neuron's not going to fire as much, just so you understand that. So now that we see these gene effects, I was like, well, now we got to repeat this study to see if there are actually any physiological effects. And so Vipa Patel, who was an undergrad doing her honors thesis with me, she repeated Liz's study, did the same sort of thing, but she then, instead of just doing qPCR, we measured body weight, weekly food intake, we did body composition using MRI, we did glucose and insulin tolerance test, and then we measured peptide hormone levels in the blood, leptin, insulin, and ghrelin, just to see if there were some of these effects. And so what we found is, to our surprise, we actually did see a physiological effect of this exposure. Specifically, males fed a chow diet, regular, chow diet, they did not gain as much weight over the 4 weeks as the oil treated. So that makes sense if we think their food intake is being decreased. And sure enough, when we looked at their energy intake, here is males, the OPFR suppressed the amount of chow that they were eating. So it's sort of like an anti-obesogenic effect. So everybody's like, oh, well, then we need to eat more of these things, and then we'll but I'm like, no, only that's going to work in males. It's not going to work in females. Right? In females, we actually saw the opposite, where the OPFR slightly increased body weight gain over the period in these overrectomized females. We didn't really see any effect on energy intake, but what we did see with fasting glucose levels, so now I'm getting more physiological and less neurological, is that OPFRs increase sort of fasting glucose in these males, but that nothing happened to females. And that's because in females, if you overrectomize them, fasting glucose is going to go up because ER alpha and estrogen suppresses glucose homeostasis, right? It controls it. So if you remove that, then you're already going to be dysregulated, and OPFRs aren't going to do it anymore. So when we looked at the peptide hormones, we saw that OPFRs increased ghrelin production, kind of suppressed it in females. Leptin, it suppressed leptin production and kind of suppressed insulin production in males too. So again, there's more of an effect in males than in females, at least the overrectomized females. But what this seems to me is like, this could be a counterbalance to the suppression of food intake that we see in the males. They're not eating as much, so ghrelin is going up because they're hungry, but they're not eating. So there's some dysregulation of the drive for feeding in the brain. This is really interesting because that means there's a localized effect in adipose tissue of OPFRs on leptin production. So PPAR gamma, ER alpha, a lot of these receptors I've talked about actually control leptin production in adipose tissue. So maybe interacting with those receptors in adipose tissue is suppressing leptin production. Same thing with insulin. So what we see here are dose and sex-dependent effects on gene expression. We see sex-dependent effects on energy balance. Males are more susceptible. They're not eating as much. They're not gaining as much weight. We don't really see much effect on females. Hyperglycemia is produced in males. There's more blood sugar. And there seem to be some differential regulation of peptide hormones. So our main conclusion is that OPFR exposures may not be obesogenic, but it's actually altering energy balance somehow, which may make you more susceptible to obesity or some other, or diabetes or some other health problem. But that's on a chow diet. So the next idea was like, well, what's going to happen if we gave them a diet-induced obesity, meaning high-fat diet? So that's what I did for the R21 experiment. And the rest of the talk we'll be talking about that. So other questions we had are why there's the sex differences, which are always very interesting to me. And what would happen if the females were intact? So for this next experiment, we did intact females, and we did the same sort of study where we exposed them to OPFRs for 4 weeks. We gave them either a low-fat or a high-fat diet, and then we did more physiological characterization of energy balance. And because we're also seeing these effects on gene expression in the arcuate, the other part of that was I wanted to see if the effects actually carry down to the neurophysiological level of MPY neurons. So we were looking at MPY activity and Ghrelin sensitivity using electrophysiology. So again, experimental design, very similar. This was conducted by 2 of my grad students in toxicology, Gwendolyn and Sabrina. So basically we did 1 dose of the OPFR mixture because I wanted to get it closer to what human exposures are. So if you're a fire person, if you're working in a plant that makes electronics, you're exposed to close to 1 milligram per day, maybe more like 100 micrograms. We're exposed to like 1 to maybe 10 micrograms a day, maybe, depending upon your environment, how much you're inside versus how much you're outside, if you're eating house dust. And then we either gave them a low-fat diet or high-fat diet, and then did body weight measurements, food intake, MRI, but then we also did metabolic measurements using clams, which is a metabolic cage, so we can get activity and food intake and water intake. We did glucose levels, we did GTT, ITTs, we did peptide hormones. We also looked at ingestive behaviors, like feeding parameters, meal pattern analysis, trying to catch as whatever we can with this sort of adult exposure model that we have. So this is a very busy slide, but I'm just going to point out the important things. So when we gave them a low-fat diet, the males, again, with OPFRs, didn't gain as much weight. Okay, that's repeated what we saw before. Low-fat is kind of comparable to the chow. It's a different type of diet. But when we gave them a high-fat diet, they actually got fatter. So in males, exposure to these compounds is obesogenic when you're on an obesogenic diet. There's more of an effect. Females, not so much. We did see a slight reduction in females this time on the low-fat diet, no effect in the high-fat diet, and that could be because they're intact. They have circulating estradiol, and I've already said that estradiol is protective against many of the effects of obesity and yes. I will get to that. Don't jump the gun. Thanks. All right. But when we looked at, so this is just sort of end time body weight. You can see this is slightly higher. This is slightly lower. Females, there's really no effect, although there's a slight decrease here. When we looked at weekly intake, just crude intake over the week per cage. So all these mice were pair housed because we don't want to have social stress be a confounding factor. So it was always 2 males and 2 females together, etc. And we dose them, just so I missed that part, is we dose them with peanut butter because mice love peanut butter. So we do dried peanut butter you can get from Whole Foods or whatever organic store, and you mix the compound in oil, and then you do it by weight, and you give them a little bit of peanut butter every day. And you separate them out so you make sure they're both eating it. But then they use the whey paper as like nesting material, so then they're setting on it and getting it on their fur, and then they're grooming it. So they're constantly being exposed to these OPFRs. So with weekly energy intake, we really didn't see much in terms of males or females. What we did see interesting is feeding efficiency. Does everybody know what feeding efficiency is? If you're in ag school, you probably do. So feeding efficiency is the amount of energy you've given and how much weight you gained. Or in some instances, if you're a dairy, it's how much energy you gave and how much milk you produced, right? So feeding efficiency is kind of an indirect measurement of metabolism, because if you're ingesting so much food and you're gaining so much weight, that's sort of a measure of how fast or slow your metabolism is. And so what we did see in females on a low-fat diet, that their feeding efficiency was lower than in the controls when they were exposed to OPFRs. When we looked at fat mass, this is where we saw the effect in males. So the males, when on a high-fat diet, were literally just fatter. That's why they weighed more. They just had more fat. So now, is this because their metabolism is slower, or because there's some effect in the adipose tissue where there's more lipogenesis and deposition of fat, right? We don't know. Lean mass was also slightly decreased, but there was no effect in females really on fat mass, as we would expect. When we looked at metabolic peptides, so here is females, males again, ghrelin, insulin, leptin. We saw effects again with males where here's oil. We know that high-fat diet reduces ghrelin production in males. It does that here with the OPs, but we did see that OPs actually reduced ghrelin this time on a low-fat diet, which is different than what we saw before where it increased it on a chow diet. So now we're having these interactions with the type of diets we're feeding them, right? So there are substantial differences between a regular chow diet and these semi-purified diets that you get from research diets, etc. And then for females, we just saw effects of high-fat diet. We didn't really see any effects of OPFR or ghrelin. Where we did see in females is we saw an increase in insulin on the low-fat diet with OPs, which hyperinsulinemia is a hallmark of sort of a pre-diabetic state, right? So that's concerning. But then when we looked at leptin, we saw an increase in leptin production in the females with a high-fat diet. So the fatter you are, the more leptin you're going to be producing. That's generally what happens. But here we see that OPFRs in the females increase. Now, if you remember in the males, it was the opposite where leptin was decreased. And here we didn't see that when we looked at these diets. So again, we're getting at this interaction of like the type of diet and the compound you're giving. Another busy slide, but what this is from the clams. This is data showing oxygen consumption, carbon dioxide production, which you can convert into RER, which is respiratory exchange ratio. So the RER is a measurement of substrate utilization, whether they're using carbohydrates or fats as their primary food energy source. So closer you are to 1, the more carbs you're using. Closer you are to 7, the more fat. So these are all the high-fat diets. So of course they're using mostly fat because they're consuming fat. And then in the males on the low-fat diet, you do see a switch during the nighttime, which is pretty standard because they're usually inactive. They're sleeping during the daytime, so they're using those fat stores, not so much from sugar. We didn't really see much effects on energy expenditure at all in the males. We did see some interactions with OPFRs in diet with water intake, so your ingestive behavior is slightly different. Wheel running and activity are just measurements, again, of activity. We saw effects of diet, so the high-fat diet ones are always less active. There's less wheel activity. They're fatter. They're not going to be moving around as much in the males. That's in the males. In the females is where it got interesting. So yay for sex differences, right? So in the V.O2, oxygen consumption, we actually saw a suppression of V.O2. Here's OP, OP's green, OP's orange, right? So it's during the nighttime, regardless of diet, there's a lower oxygen consumption in these females. Same thing over here where you have changes in how the substrate utilization occurs. So we have some differences here at night. Again, with energy expenditure, you have some nighttime when they're more active, some changes in expenditure. Water intake was not directly affected by OPFRs, but what happens is you didn't have this pattern. So it's a pattern effect where OPFR exposure eliminated the diurnal cycle in water intake in females. But this is where it got really interesting to me and totally shocking. I had to make sure everybody checked and triple checked their data because we saw OPFR exposure, regardless of diet, reducing activity in the females. So this is only overexposure by now like 6 weeks, and they're just not moving as much. So what would happen if we did it for 6 months and they didn't move as much? Maybe they would have switched over to actually getting fatter because they're just not moving as much. But it also correlates with their less oxygen consumption. So there's a switch between activity, suppression of activity, suppression of oxygen consumption, metabolic rates. Now a lot of that is controlled by the brain. Activity is controlled by the brain. I'm not saying it's controlled necessarily by these hypothalamic neurons I'm looking at, but there are effects happening in other centers of the brain that I'm not studying that these OPFRs are reducing. So one of the things we're doing now is looking at open field tests, elevated plus maze, all those sort of anxiety, locomotor behavior assays to see is this associated with some sort of neurological effect. Yes?
Sort of related to that, is I was curious about the anxiety behavior. Because one of the gene expression changes you described is Pbdes. Obviously, there's lots of different
Yep. Products.
peptides or
Yep.
products
Beta endorphin, blah, blah, blah.
Endocrine stress, like
Yep.
ACTH and port. Have you ever looked at stress response, HPA activity?
No. But it's what you always propose to do in a grant, and until you don't get the money, you don't do it. It's one of the things I'm like, yeah, let's look at this because So
I'm just
definitely
thinking
some.
that, especially once you start doing those anxiety tests, having
Yep.
both of that information
Yep.
help figure out.
Yep, exactly. So again, love the sex differences. This case, females seem to be more disrupted than the males sometimes in some instances. So when we looked at meal frequency and meal patterns, in males, we saw no effect of OPFRs, only effects of diet. But in females, we actually saw an effect of OPFRs. And here it's because they're not eating as often on a high-fat diet when they're exposed to OPFRs. So maybe the reason why they're not getting fat, even though they're not moving as much, is they're not eating as much. Like their actual meal pattern is changing by OPFR exposure, and that kind of counteracts what we're seeing with their metabolic. So meal size is pretty much no effect. Same thing with duration. It's really basically how often they eat is changing. They're not eating as much when you're on a high-fat diet. And again, that's controlled by these POMC and MPY neurons that I mentioned. And lastly, I'm looking at sort of peripheral stuff because I'm becoming more of a holistic physiologist and not just worried about the brain. So we did glucose and insulin tolerance tests. We really didn't see any much effects in males. This is diet. This is all diet. Although the OPFRs reduced glucose intolerance, or basically increased the amount of glucose that was shuttled from the periphery into muscles, went on a high-fat diet. So diet-induced obesity is known to induce glucose intolerance. OPFRs may actually help against that. I don't want to say that they're protective, but it does look like that. Females, we didn't really see very much either. So to me, this means it's mostly the effects we're seeing are central. They're not happening in the liver. There may be some effects in the fat because that's where we're seeing the effects of production of leptin. There may be some effects in the gut because you're all changing how ghrelin is being produced. But most of the effects that we're seeing are pretty much central. So again, sex-dependent effects, which are always very interesting. This time males got fatter, but as we physiologically characterize the animals, it seems like the females were more susceptible, especially with feeding efficiency, meal frequency, and activity. Again, there was some alteration in the peripheral hormones, but it did not affect glucose homeostasis. So what we're now interested in is characterizing the neurophysiological effects, and this is what my grad student Gwen is continuing on for their thesis. So in that study, we wanted to see, because we saw effects on MPY expression and AGRP expression, and we also saw effects on KCNQ channel expression. I have several papers showing that KCNQ channels are in MPY neurons are regulated by fasting, by estradiol, by high-fat diet, by ghrelin. So really characterized it well in the system. And so what Gwen did was took intact adult male and female mice that are MPY GFP. That means they have the green fluorescent protein tagged to the MPY promoter, so you get GFP in MPY neurons. So we could target those neurons specifically and expose them to the OPFRs. First, we did single-cell harvesting to then look at gene expression specifically in MPY neurons, and then we followed up that with electrophysiological examination of the M current. So just to give you an idea of what I mean by cell harvesting is we took the treated animal, decapitated it, did some coronal slices, and then cut out just this area of the brain, the arcuate, right at the base of the hypothalamus, titurated in proteases, and sort of dispersed the cells onto a little petri dish with had a glass slide, put it in an inverted fluorescent microscope, shone the light through the slide, and we saw the green glowing neurons that we wanted and ignored the other ones that didn't glow green. Collected them. We either did single cell or we pooled them into pools of 5. The pooled cells we used for quantitative real-time PCR. The single cells, we just did regular PCR to look at just individual co-localization patterns of different types of the receptor for ghrelin as well as the KCNQ channels. So straight up gene expression, this again is in pools of MPY neurons from females that have been treated with OPFRs or oil. And what we saw, again, in the adults, in the wild types, before we really didn't see much effect in females in MPY and AGRP, but we did see an increase in KCNQ2 that was approaching significance. It was very disappointing to see 0.06, but it is what it is. This is significant, so you see an increase in ghrelin receptor. So GHSR is well characterized in males in MPY neurons, and I've been doing some characterization in the females, but it basically, when it's activated by ghrelin, it turns on MPY neurons and then it drives hunger, right? So if they're expressing more GHSR, maybe they're more sensitive to ghrelin and they can be more hungry. But if they're expressing more KCNQ, there's more M current, the neurons are not going to be as excitable. So it's this sort of interesting balance. Oh, by the way, so in males, when we did this, we saw suppression of MPY, and then there were 2 of the channels that were, again, like 0.06, 0.07. It was really disappointing. All right, so to do the electrophysiological studies, of course, we use these green fluorescent proteins. So these are the MPY neurons with the GFP. They're tightly packed around the median eminence. Here's the median eminence, main part of the arcuate. So just to give you an idea of what the M current does, it controls the refractory period. So after a depolarization, repolarization, it sort of extends the refractory period. So firing frequency is altered, it's extended. It also can, so it's part of the after hyperpolarization current. It also can change the resting membrane potential if it's strong enough to make it more negative, right? It's a potassium current. And here's an example of an MPY neuron where you gave XE991, which is a potassium current inhibitor, selective KCNQ inhibitor. Sure enough, if you give XE991, eventually the neurons are going to depolarize enough that they're going to start firing. Right? So what Gwen did is measured the M current. We did these sort of deactivation protocols. So here's the M current IV plot with oil and with OPFR. And actually what we saw in these females is like there seems to be a slight suppression of the M current. So that makes the cells more excitable. And what we saw is like the peak, again, all these 0.056s and 0.06s is really getting depressing after a while. But we're excited by it, right? It's still, there seems to be something going on. So here's the max current is suppressed with OPFR exposure. The really, again, input resistance is another measurement of total activity of potassium currents in these neurons. And we did see an increase, slight increase, again, almost significant, with OPFR treatment. That means there could be more of these channels that are open and active at that time. And when we looked at resting membrane potential, they were actually, the change in resting membrane potential with the M current, with the blocker XE991, we did see a greater effect in the OPFRs. So that means there's more KCNQ channels, you block them all, there's going to be more of a change in the resting membrane potential. Finally, when we looked at the Ghrelin sensitivity, so here is 2 different concentrations of ghrelin, 100 nanomolar and 1 nanomolar. 100 nanomolar is what most people use when they do electrophysiology with ghrelin in any neuron in the brain. So we wanted to do a small dose response with that. And what we saw were changes in the current that were, again, almost significantly different. Again, almost significantly different between oil and OPFR in terms of firing frequency. They seem to be more excitable. And then when we looked at the change again in resting membrane potential within these 2 concentrations, again, this actually it was like a shock. Oh my God, something was actually below 0.05. We actually had toasted some tequila for that because it was so frustrating. You got to celebrate the small things, right? So we saw an effect that basically this means that they're more sensitive to ghrelin than when they're on OPFRs. So if they're more sensitive to ghrelin, potentially these females are going to be eating more, right? But what we saw was that their meal duration was suppressed, but there wasn't a change in the meal size. And so what we're thinking is actually it's a change in the meal rate, meaning how fast they're eating, right? So they're eating faster when they're on OPFRs than when they're not, right? So what we're doing now is characterizing OPFRs in POMC neurons. That's the last chapter of Gwen's thesis. And right now they've done the qPCR, or doing the qPCR, looking at leptin and insulin receptors, some KCNQ channels. Leptin and insulin act on TRPC channels in POMC neurons, so we're measuring TRPC5. And then they're going to be doing some of POMC excitability and hormone sensitivity, just like with the MPY nodes. So that's sort of the direction we're going in now. So because we think there are some effects, these are subtle effects, but there is this pattern of a change in the neurophysiological properties and hormone sensitivity to these neurons when they're exposed to OPFRs. So what we're doing now is looking at the effects of OPFR exposure in ER alpha knockouts and in brain-specific PPAR gamma knockouts. So PPAR gamma hold knockouts are fetally lethal, so you don't get them, they don't live. So you have to do tissue-specific. And so we're using SYN1 Cree with a PPAR gamma flux and getting these brain-specific deletions and then repeating all this physiological examination. We also are doing some maternal stuff, so I've got a lot of really whole nother talk on all that. Some really interesting stuff showing again some of the effects on energy homeostasis, glucose, cardiovascular effects. Maternal exposure to OPFR increases blood pressure in males only, but not females. Some effects on glucose, so there could be sort of a metabolic syndrome effect because these animals don't necessarily need to be fat. They can be thin and have these disruption to metabolism. So yeah, that's kind of where we're moving next. I want to thank Elizabeth Crum and Vipa and Gwendolyn and Sabrina and my lab manager Ali, who sort of controlled everything that was going on in the lab, and all my funding sources from NIEHS and as well as Rutgers. Thank you.
Time for questions. Is there any questions? So I have a question
Yes.
to start off with. So you had shown the in females given OPFR, they have higher expression of one of the KCNQ channels.
Yes.
And then the other thing was the ghrelin receptor.
Yeah.
But their end current was lower.
Right.
Why is that?
So
I don't understand.
we don't understand that either.
Okay.
So
I thought maybe
one
it was-
thing that it didn't come up in terms of gene, so what I didn't go into more detail is the M current is really heterodimers of KCNQ3 with 5 or KCNQ3 with 2. So while we didn't see any effects on 3 and 5, I've already shown that fasting suppresses 3 and estradiol increases 5. So the M current's very complex in these MPY neurons because it depends on how much 5 or 3 or how much 2 and 3 you express, you're going to get a totally different M current in each neuron. So a lot of the studies I do, we have to do like 20 cells just to get some sort of effect, right? Mm-hmm. So with that, thinking the M current is lower, but we know I have papers showing that ghrelin inhibits the M current in MPY neurons. So that's the connection.
That's where the ghrelin receptor is high, so therefore it's more sensitive to ghrelin.
Yeah.
This is really tough because you have all these factors coming from the body acting on the same
Yes.
neurons, and then you have nearby neurons also influencing them. They're also influenced by those same factors, but in an opposite way.
Right, so that's why things aren't easy.
No.
And why we get a lot of .06s.
Other questions? Yeah.
So KCNQ 2 and 3 are both involved in vesicular release. Is there a difference like in the mean rates or
In what rate?
mini potentials?
Mini potentials. We have not really looked at that. that's something easily done. We could do that, but no, we haven't looked at EPSCs or anything like that. Yes.
Question, I might be forgetting it, or maybe I missed it. So do the OPFRs, do they actually bind to the estrogen receptor? Is that
So they have been shown to, well, they say interact, but they bind. There is binding affinity. It's much lower than estradiol.
okay, but that's the receptor you think.
It's either that or PPAR gamma could be one. The reason why is because we saw changes in gene expression with ER alpha and PPAR gamma, right, in the initial study, but we saw nothing in the females with ER alpha. And so that makes me think it's more of PPAR gamma, because if ER alpha was activated by OPFRs in the females, they would be, ER alpha expression would be downregulated, and that didn't happen.
And my related question is, because I'm thinking about the sex effects, if you just think about those, whether it's the estrogen receptors or the PPAR alpha, what are just the known baseline differences between males and females? Like just, you know, if
Oh,
they're more or less than one or the other.
oh,
or
so-
different isoforms, just I'm thinking
sure.
the baseline's different than their.
So there are big differences in ER alpha between male and females in the arcuate. Females express much more. I mean,
So I'm just wondering if that's somehow.
right, and that may be related to some of these effects. Now, PPAR gamma, I'm not sure. I don't think anybody's looked at sex differences. We usually could. We have the qPCR. Yeah, no, it's easy enough to do. Something we can do to compare, but we have not actually looked at that. So that,
We would look at that.
yeah, because
We already see it in NAC. So.
yeah, you see in the NAC, the PPAR gamma. So it's interesting because now that again, you never know what you're going to do, but now I'm reading all these papers on PPAR gamma over the past couple of years, and you know, we are seeing now effects when our brain-specific knockouts on a whole host of activity and behaviors, OFT, EPM, activity, aggression. If you take two brain-specific, you know, PPAR gamma knockout males who've never seen each other, they go like, oh, hi, and they, you know, they cuddle. They don't fight, right? So clearly there's a lot going on with PPAR gamma in itself that is totally sex-dependent. And, you know, that is something that certainly, it's what I propose to do in these grants that get scored but not funded. So let's just say, I think about it. Yes?
I don't recall any of your qPCR data if you looked at any astrocytic markers. I know that in that area there's some signaling of, I might be wrong, alpha MSH on astrocytes. So I was wondering.
Oh yeah, there are. There is. I have not done any astrocyte stuff or glial stuff. It's certainly something of importance because they do express these receptors and they probably are involved in some of these effects. I just never not gone that far. Yeah. Yes.
This is a question more about the peripheral, but so you suspect that there's a difference in feeding rate. Did you look at either assimilation efficiency, so how much is actually, you look at feeding efficiency, but how much is actually being assimilated from the gut, or did you look at any?
Oh, you see, so looking at like their fecal, no, we have not done that.
Looking at potential like uncoupling of mitochondria or any other explanation of like.
So there is some evidence that some of these OPFRs can interfere with mitochondrial function, not the ones that I looked at, not the ones that I used. So that is certainly somebody who is a cell biologist who can do that, would be of importance, but it's not something I'm directly, but it would make sense with the substrate utilization changes that we see, that there is some sort of mitochondrial disruption because there are changes in how they're using carbohydrates versus fat. Yeah.
Other questions? Okay, great.
Great. Thank you.
Lunch, for those of you who signed up, there's lunch afterwards at Sandy Hall. We're going to be walking over there. Great, thank you. Thank you very much. Yeah, awesome.
Thanks.
Thanks. And thank you for accommodating with the
Yeah, the
technical
drama.
difficulties. Yeah. I knew once we got the projector going, we'd just be like,
You'll be fine.
we're good to go.
Yeah.
So, I mean, do you think that OPFRs are acting on estrogen receptor in males? Because you see a big change in expression there.
Yeah, you see change in expression.
Yeah.
They could. I mean, ER alpha is expressed in males.
Yeah,
I mean,
it's just lower, but you know, it plays an important role.
yeah, it does,
You
especially
give-
with
you give-
reproduction.
Yeah, you get estradiol.
Because a lot of, you know, like
that changes things for
negative feedback is through ER alpha signaling in males even.
Uh-huh.
So, yeah, I mean, there's a lot unknown here. So these are questions I want to answer, but when I, you know, trying to get money. Yes.
Yeah. Okay. First of all, I love your jacket.
Thank you.
Second, did you say something? I think you had on a slide that you were doing qPCR with 5 cells.
Yes, 5 neurons. Yeah, so we do single cell harvesting, but we pool 5 cells together and we do qPCR on it.
Is that really enough? Like, you can really get enough cDNA from 5 cells?
If they're genes that are highly expressed, like the neuropeptides
Yeah, I guess that makes sense.
and the casein Q channels, we have no problem. We have some problems with like insulin receptor, but I think that's a primer problem more so than expression.
Gotcha.
But you can do RNA-seq on one cell if you do amplification and blah, blah,
But
blah.
you don't have to do amplification.
No?
Just standard.
5 cells. And if it's,
Yeah,
it's
extraction.
something you just
makes
got to
sense.
have the right
Yeah.
equipment to get the cells
Yeah.
and get them healthy and nice,
Right.
and then you just do regular RT-PCR, do the RT and then you do the qPCR.
As long as it's expressed highly enough.
Yeah, and we usually do like 3 pools per animal for each gene, and you
Gotcha.
can get like 2 target genes and 1 reference gene from each pool.